Yes, indeed, my name is Jeroen van Zon. I'm a group leader at AMOLF across the street. And what I want to talk about today is something that in our group is really a new direction, namely organoids. So the same organoids that Ina also teased with at the end of her presentation. And what I also want to do here, given that we're also here sort of to talk about the future, is to make a case that these organoids also really represent an exciting new opportunity for the fields of quantitative developmental biology, physics of life, to push these fields really in new directions. And before I tell you what I mean by that, I first want to give a little bit of background of my group and how and why organoids entered it. So as Craig mentioned, so I'm a physicist studying developmental biology, and the research in our group is really driven by this sort of central mystery that is highlighted here, that was also alluded to yesterday in some of the talks. Namely, that on the one hand, we know that all our cells on the single cell level are really strikingly variable, for instance, in terms of gene expression. Yet somehow, when they come together and build bodies, they can make these incredibly mind-bogglingly complex structures, but they do so seemingly without error, and even better, they do it to almost identical specifications. And so we want to understand, how does this happen? And typically, when people study development in a quantitative manner, the way they do that is by using simple model organisms. And in a way, our group is not different. So in the sense that half our group is working with this model organism, C. elegans. It's a small nematode worm. And a typical experiment in a lab that we do is that we have these microchambers. We load a single C. elegans egg in there. It hatches. There's a little baby worm. And over the course of 2 days, it grows into an adult worm. And what we can do now is at each time point, we can go into the images that we take of this worm. We can find the cell that we're interested here. For instance, we can recognize it by a nuclear fluorescent reporter. And then at each time point, we can see what that cell is doing, and then we see it every once in a while that it divides. And in that way, we can build these cell lineage trees to study how development happens here. And so at this point of my talk, I could easily also have given a very different talk about how amazing C. elegans is as a model organism to study development. But for the purpose of this talk, I actually want to highlight rather what people often perceive as the limits of this type of work. So one clear limit is that these worms are quite far removed from us evolutionarily. And so whenever you learn something about development of C. elegans, you always can ask the question, okay, so how relevant is it for us, in the sense that they lack many of the cell types or tissue structures that are really central to human biology. There's also another kind of criticism, and that is perhaps even worse, and it is that these organisms are so far from us, so simple, that they actually even become a little bit boring. And the way in which you can argue that C. elegans is a bit boring for people like me who are interested in variability is that if you look at exactly the same cell, but now in different animals, you will find exactly the same lineage tree. And this sort of invariance of lineage trees from animal to animal actually holds for all cells in the worm. So in that sense, there's very little variability on that level. And so I always thought it would be really cool to take these sort of quantitative measurements and use them to study cell dynamics in a system, a developmental system that would be much closer to us, to humans, but also have more variability, for instance, in cell lineage. And one such system that fits this bill very well was already introduced briefly by Ina, and that is our intestine. So this is what the lining of our intestine looks like. So you have these finger-like structures called villi, and they're lined by a single layer of cells, and most of these cells are actually involved in absorbing food from the intestinal lumen. But what is striking about these cells is that they only live in our bodies for 3 to 5 days, and after that they are renewed. And they're constantly renewed by a small population of cells that sit in these hidden structures here called crypts. And they renew all these cells. So if you would look at this in more kind of an idealized textbook-style picture, this is what people typically show. And what you can see is that there are different cell types and that they occur in very distinct spatial patterns. So the stem cells here in red are sitting at the bottom of these crypts. Above them, you have this group of cells that are still dividing, but not yet differentiated, so they're in the process of differentiating. And they're called transit amplifying cells. And above there, when you enter the villus region, you'll only find differentiated cells. Most of these are these light blue cells that are absorptive cells, so these are the cells that absorb foodstuff. But interspersed between them, there are also these secretory cells. They occur in different types, so most of these are actually called goblet cells, secretory types that are goblet cells. They produce the mucus layer that coats the lining of our intestine to protect it. But there are also other types of secretory cells, like enteroendocrine cells, that produce hormones that are really important for our appetite and digestion. There are also special secretory cells. They're special in multiple ways. One is that they're not here in the villus, but they're actually sitting here intermingled with the stem cells. And they're also special in the sense that we now know that they secrete all sorts of factors that actually form the stem cell niche. So cells that are sitting next to a Paneth cell, they tend to remain stem cells. And at the same time, because of this proliferation of cells here, there's this idea or we know that there is this sort of a conveyor belt type motion. Cells are born here, they're being pushed upwards by more cells being born here. They travel up to the tip of the villus, and there, at the end of their short lives, they are shed into the lumen, and their job is over. What is not so clear, if you look at these type of textbook-style pictures, is that this process is not only highly dynamic, but it's also really intrinsically stochastic, meaning that all the key decisions to remain a stem cell and proliferate or not, to differentiate, and if you differentiate what type of cell you will become, all these decisions are inherently stochastic. And so a really important question that you could ask, given all this inherent variability, is how does this tissue now ensure that all these different cell types are produced and maintained during its entire lifetime in exactly the right proportions?
Sorry, just to interrupt.
Yeah.
How do you know they're stochastic?
Can I come back to that at the question and answer session at the end? Perhaps then it's already a little bit more clear. Yes, so if you are interested in this specific question, there are certain technical challenges that you have to surmount. And so what sort of approaches do people now use? What is sort of the current state of the art if you want to address this question? So one challenge is that these cell lineages, how you go from stem cell to differentiated cell, are actually really complex, much more complex than the lineages that I showed you for C. elegans. And one way in which people can probe that is by this technique called lineage tracing, in which in individual cells you induce a heritable marker that is then visible in all its offspring, as shown here, and here for multiple colors in sort of newer versions of this technique. But they have one major downside, and that is that these images, you typically get those as single snapshots. And it means that, yes, you can see that all these cells here were offspring of a single cell at some point earlier. You really miss all the dynamics that lead you to this offspring. So you're really blind to that. The other challenge is the relatively large number of cell types that you have to consider all at the same time. So in the intestine, the number of cell types is at least 7, although you can arrive at a higher number depending on how you lump or split. And the current state of the art here is to do single-cell RNA sequencing. So this is an example for the intestine. So each of these dots is a single sequenced cell, and the color indicates what type was inferred from the gene expression. And you can see many of the types that we know in here. Some of these that are here pulled together, they're actually thought to be different stages in differentiation of the same cell type. So this type of sequencing gives you some idea of time, of dynamics of differentiation, but it's really quite limited. And really a big limitation is that there's really so all these cells come from the same tissue, but by the time you sequence them, you really know nothing anymore about their spatial relationship. And given that these cells respond to complex spatial signaling cues, but also communication with their neighbors, this is really something that is extremely important if you want to understand the differentiation. Perhaps the most formidable challenge of all is if you want to see this type of differentiation dynamics, is that it's all playing out hidden deep inside bodies. And so it's very hard to follow this. This is a difficult problem in mice, but it makes it essentially impossible to study in humans. And so this is where organoids come in. So organoids are small mini organs that can be grown in the test tube outside of the body from single stem cells, small bits of tissue. And this here is an intestinal organoid, a mouse intestinal organoid. It might not look to you a lot like the intestine picture that I showed you, but you can see these parts with very distinct morphology that actually do correspond here, this to the crypt region, and this is sort of a hollow sphere that corresponds to the villus region of the intestine. What is really nice about these organoids is that they really display the full range of cell types that you see in vivo. And what is even something to note is that many of these cell types are actually very relevant for understanding disease. So they can be functioning not properly when you're diseased, and so they're really medically relevant. And the only way for many of these cell types to currently culture them in the lab is in the context of these organoids. The other thing that I really like about organoids is that they're very accessible. You can put them under the microscope, and then you can make movies like this. This is an organoid grown over the space of 2 days. Each of these blobs here is a single cell nucleus, like I showed you in the example of the worms. And you can see that they're very dynamic, they move around, they divide a lot, and this is something that plays out in vivo, but you cannot see it very easily. Here you can very clearly see it. And again, to make the point, this is mouse intestinal organoids. Everything I will show you is about the mouse intestine, but these types of things you can also do for human organoids, even from tissue that you can get directly from patients. And finally, this is perhaps a little bit of an aside. Something that really strikes me about organoids is that these sort of shapes and cell type patterns, they're all generated without any of the cues that these tissues would normally encounter within the body. And what I find striking about this is it kind of presents a slightly different picture of development. In the sense, people typically think of this, that you get these patterns because there are these very precise cues coming from the rest of the body that do the patterning. But this shows that a lot of development is actually a form of self-organization. And how this self-organization arises and how it interacts with development inside the body, this is really a completely open question. So coming back to this organoid, so we wanted to know, given that you can image these now, can you now follow all the relevant dynamics for differentiation? Can you follow that now in all of these cells in the organoid? So things like cell lineage, cell type. And what I should specify here is what I mean with we. So this is really a joint venture, you can say, between my group and the group of Sanne Tans, who's also here in the audience. So all of the work that I will show you here is work that we very much did jointly. And so the question that we were faced with when we were looking at these organoids is, can you track cells in there? Can you follow lineages? And briefly, I can say the answer is yes. If you want to see more details, this is a paper that we published on the technique. The idea is that we use neural networks to predict, based on these confocal images, where the center of a single nucleus is. And you can see from these movies here that this works really strikingly well. I'm very impressed with these neural networks. We then have, for each time point, we have a cell, a nucleus of the center of a cell nucleus. We then use a technique called a min-cost flow solver to link a cell at one position with the same cell at the next position. And we also use this when a cell is dividing to connect the mother cell to the two daughter cells. And finally, what we do is that we also, when we make these data sets, we go past each division that we identify individually to just see by eye that we are making no mistakes, because that would really impact these lineages that we get very strongly. And so the nice thing about this is that now not only can we track these cells in these organoids in time, but we can now also measure, purely by cell tracking, we can measure these cell lineages. We can see how these cells are related to each other. So this is what you get if you do that. So this is again the organoid that I showed you before. This is the reconstructed organoid that we get. Each of these white spheres is a single cell that we have tracked. And the colors now represent cells that are offspring of the same, of one cell at the beginning of the experiment. And so you can see, for instance, here this yellow lineage corresponds to all these yellow cells here. They were all offspring of a single cell that was there at the beginning. You can see that some lineages divide very little, some of them even don't divide at all, and other ones divide a lot and are presumably stem cells. What is nice is that you can also see dynamics, interesting dynamics within a lineage. So you see that this cell divides once, twice, thrice, and then here, these cells, they stop dividing and presumably differentiate, while these pair of cells keep on dividing and are presumably something like stem cells. And so we used already data like this to try to understand how proliferation, cells continuing to proliferate and cells that stop proliferating, how that is balanced within these organoids to ensure that the number of proliferating cells doesn't explode or doesn't dwindle to nothing, but stays at a relatively constant state. So I showed you that organoids they kind of solve this problem of accessibility. You can look at these processes that are normally hidden within our body. I also showed you how you can use cell tracking to measure these complex cell lineages. And there's still sort of this remaining problem, and it is that there's large number of cell types that are sitting in these tissues that you want to follow if you want to understand how they're specified. And this is what I want to talk about in the remaining part of this presentation. So the way we solve this is as follows. So we start with what we already know how to do well, so this is time-lapse imaging. So we start with time-lapse imaging of a growing organoid. Then we use the approach that I already outlined to track all these cells. So we know exactly all the cells at the beginning, and we know all the cells at the end, and we can connect them in space and in lineage. But now what we do is, at the end point, we do different rounds of antibody staining. And each time we do that, we can stain for one of these 7 cell types that you find in the intestine. So this is an example of what this looks like for this crypt here. And here we use an antibody orphan-4 to stain stem cells. And so here you can see that there's a group of stem cells. Wheat germ agglutinin is something that binds to the mucus that is produced by these goblet cells. And so here you can see a mucus-producing cell. Here a Paneth cell by lysozyme, and here you see these absorbing cells, enterocytes, by staining what is aldolase B. And in that way, what you can do now is that you have your tracked organoid at the endpoint, and now in a way you can color all these cells that you see here by the cell type that you get from the antibody staining. So here at the end of the crypt are all these stem cells, same here. They're interspersed with different kinds of secretory cells, and here you see lots of these absorbative enterocytes. And what is really nice now is that you can go back to these lineages, but now you know at the end also exactly what the type is of the cells that are generated there. And there are a couple of interesting patterns that you already see at this stage. For instance, that you see that stem cells, and often also these absorbing enterocyte cells, they occur, and also they occur in larger clones, meaning that you have many closely related cells that were generated by one cell that all have that specific type. And we also find that there's a group of cells that are not labeled by any of our antibodies, that we for now call unlabeled cells, that also exist in these larger clones. Oops. We also find that there are some cells that are sitting inside the crypt, you can see one over here, that don't divide at all. And what we indeed see, which is consistent with what we know, is that these are Paneth cells. So these are these secretory cells sitting in the crypt that make stem cells. And finally, something that we found quite striking and unexpected is that we see that very often, almost always, sister cells have the same fate. This is not so strange perhaps in these larger clones, but if you look at secretory cells, like here these mucus-producing cells, we see that they're always born in sort of doublets, 2 sister cells that have the same fate. And this is actually quite surprising, because the idea is that these secretory cells, once they're specified as a secretory fate cell, they differentiate and that they stop dividing. So it was quite unexpected that we see this here. So we quantified this more carefully as follows. So here what I show you is we look at pairs of sisters. We look at what one sister is, the type, and then we look at, for all sister pairs, at what now the probability is that you see the other sister with one of these types shown here. And what you can appreciate, presumably, is that all these points are very strongly concentrated along the diagonal, meaning that if you look at sort of the global level, indeed you see that sisters typically have the same fate, as I said, which is quite unexpected. So what you can also see is that there are some exceptions here, so they're quite rare, but you sometimes see, for instance, sisters where one is clearly a stem cell and the other one is unlabeled. So we see some cases where sisters are slightly different. And you can look at that by zooming out a little bit. So one analysis that we did is that we looked at larger lineage trees. So for instance, a lineage tree like this, and we selected trees like that with sort of the constraint that all offspring could only be of 2 different cell types, sort of like 2 cell type lineage trees. And now you can ask yourself, what pairs of cell types do you find together in one tree? And the reason why that is interesting, because it implies that there was sort of a choice there. There was at one point a choice to go this direction or that direction. And by mapping what sort of types of, what sort of pairs of cell types you find together, you can get sort of an idea of the available space of choices that is there for these cells. And if you do that, you get numbers that are quite a bit smaller, but still you see very interesting patterns. So again, here, this is, if you have a lineage tree like that, it would be that one part of the lineage tree is an androcyte, the other one is unlabeled. And if you do that, there are a few things that are striking mostly because you don't see them. We never see any lineage trees that have a combined absorptive cell type or secretory cell type. So we never see that. The other thing that we never see, sorry, seems to be a bit stuck here. What we never also see is that we get lineage trees that have mixed secretory types, that have either goblet cells or mucus-producing cells. And the way we think about this is that this implies that there is not sort of a secretory progenitor cell, so one cell type that is destined to be a progenitor cell and gives rise to different kinds of progenitor cells. And this is something that people have speculated about, that this exists. Sorry. So, yeah. I solved it, I think. And the other thing that you see is that that actually many of the differentiated cell types, from enterocytes to enteroendocrine cells, they're typically generated in lineage trees where the other cell type is either stem cell or unlabeled cell. So somehow these stem cell and unlabeled cell types seem to be very close in the lineage trees to these differentiated cell types. So it seems that they're somehow special. We can look a little bit in how they're special by looking at something else, and that is the age of these cells at the moment we see them at the end of our experiment. So here you have a lineage tree that we measured, and we know that they have a particular cell type at the end, and now we can calculate the age of the cells at that time by just looking at the time between the end of the experiment and when they had the last division. And when we do that, we see that for almost all these differentiated cells, that age is relatively large, so it's somewhere between 20 and 40 hours. But what is interesting is if you look at stem cells, that they appear to be much younger in the experiment. So we see, if we find at the end of an experiment that the cell is a stem cell, the time since the last division was typically rather short, so 10 hours or so. And what was interesting is that these unlabeled cells, so these cells that are not stained by any of the antibodies that we used, we find it has a similar young age. And these stem cells, they're of course always proliferating, and this explains why they have such a young age. And this suggests that these unlabeled cells that we see here are also rapidly proliferating. And so that, together with other evidence, allows us to identify these unlabeled cells as these transit amplifying cells. So cells that just stop being stem cells, but have not yet committed to any differentiated fate. So all the analysis that I presented so far is analysis that is purely based on the knowledge of the cell lineage and what cells are at the endpoints. But it means that in our experiments we also encounter many cells that are not present at the endpoint because they have divided, and where we have no measurements of what cell type they are. And so one thing that we've been searching for and is a way to assign, based on the endpoints and the lineage, to assign cell type to these earlier cells. And this is what I want to show you here, and it's sort of based on the results that I presented in the previous slides. So imagine that you have a lineage tree like this. So at the end you have 2 unlabeled cells and 2 stem cells. So first I showed you that often these sister cells have the same fate. And the way we interpret that is that they have the same fate because that fate was already set in the mother and that mother divided again. And so if you want to trace back in time, we have one rule that we use, namely that if you have 2 sisters of the same fate, then we assume that the mother had that same fate as well. So this works well, but at one point you run into a problem. You run into the problem, in this case, for instance, where this sister has one fate and that sister has another inferred fate. And now is the question, what would that mother cell then be? And here we make use of the fact that we know that stem cells are at the top of the differentiation hierarchy. So a stem cell is only produced by division of a stem cell. And so in this case, we use the rule that if you have one cell of a stem cell type and the other one of a different type, then the mother must be a stem cell. we also often encounter lineages like this, where you don't have any stem cells, but you have here, for instance, 2 differentiated enterocytes and unlabeled cells. Here you can again propagate back using that simple rule of sisters, but again you get a decision here, and here we add another rule for this backtracking procedure, namely that if you have one unlabeled cell, which we identified as transit amplifying cells, and the other is differentiated, then the mother must be a transit amplifying cell as well. And you can ask yourself, how many more rules do you need in this case? But what I showed you before about these 2 type lineage trees basically means that then you're done. You know that there are no other choices that these cells in these lineage trees were faced with. And so with these rules, we can propagate every lineage tree back. The question is now, how well does it work? So these are the results that we get if you do that. So now here, each number corresponds to the rule that we apply to infer that cell type. We can, in this way, as I said, almost all the lineage trees that we get, we can infer a cell type here. But now we can look back to the movies and we can ask ourselves, okay, what does it look like? And what you find is, at least there, it makes a lot of sense. So what we see is that stem cells are typically always, inferred stem cells are always found at the tip of the crypts. On the other differentiated cells that we see there are inferred secretory cells. We see that there's a regime of inferred transit amplifying cells that separates the stem cells from a regime of inferred absorptive cells. So at least in this way we can reconstruct the correct spatial patterns. So something that is really nice about this method is that it also tells us where changes in cell state, in cell type are. So here there are cases where the mother had one cell type and the daughter have different cell types, and so something changed there in these cells. And so it tells us where differentiation is happening. And so one question is, can we now use these sort of inferred cell type transitions to also say something new about differentiation? So at the very last part of my talk, I just want to illustrate briefly how we can say something about this. So we looked at a number of things, but one thing that we looked at as well is the relative ratio of absorptive cells and secretory cells. This is one of the earliest choices when a cell starts to differentiate, is whether it wants to become a secretory cell or an absorptive cell. And it's regulated, and what I should say is that if you look at the final patterning in the tissue, is that you always find these secretory cells isolated, so they're surrounded by absorbing cells. And what people know is that this decision is controlled by notch signaling, so the notch signaling that Ina just mentioned. And the idea is that a secretory fate cell is producing a ligand, delta, that then can bind to notch receptors in neighboring cells, and in those cells, inhibits secretory fate and instead stimulate absorptive fate. And the idea that people have had is that this kind of naturally explains these 2 facts. So the idea is that at the beginning of differentiation, there's upregulation of delta fate, secretory fate, in all these cells. But by notch signaling, you end up with one secretory cell inhibited, surrounded by a much larger number of absorbing cells. So notch signaling in this way could explain both the difference in ratio and sort of the spatial pattern. So what we did now is that we went back into our data and we asked ourselves, okay, can we see signatures of this? So we have these lineage trees, and here we can see, for instance, a transition to absorptive fate. Here we can see a transition to secretory fate. And one thing that we asked ourselves now, do we find that transitions to absorptive fate happen more frequently than those to secretory fate? And what we find is that this is actually not the case. So the thing to look at are these here, the transitions per hour to all secretory fates and to the absorbing fates. And we see that these are quite similar. And so it means that these transitions to secretory or absorptive fate are made at the same rate. So something else must be changing to explain this difference in abundance. And here we can look at another aspect of this, namely cell proliferation. So what I plot here for different cell types is how many divisions they typically go through since the first time that we see them in the lineage. So for instance, here we see transition from stem cell to transit amplifying cell, and then we see one two divisions there. Indeed, when you look at this for stem cells, we see that they divide a lot. So we see many divisions. For transient amplifying cells, we also see that some of these lineages have very many divisions. It's quite different if you look at secretory cells. So there we see that most of them have one division, sometimes two. For Paneth cells, very rarely three. So the number of divisions that we see for these cell types is much larger. And what is then interesting is if you look at enterocytes, there we see that the number of divisions is quite a bit larger. And so what we think is happening is that these lineages that give rise to secretory or absorptive cells are generated at the same rate, but that these absorptive cells, they proliferate many more times after specification than the secretory cells. And that is what controls the abundance. The final slide that I want to show here is about the patterning. So we know that, that in the tissue itself, secretory cells are typically surrounded by the absorptive cells. But there's sort of a bit of a mystery in our data, namely that most of the secretory cells that we see are born as sisters. And so they're almost by definition sitting next to each other. And the question is now, what happens next? And so one type of analysis that you can do is that you can look at neighboring cells at some time, and then you can look at the later time and see what has happened. Are they still neighbors or are they separated? And if we do that for neighbors that are not sisters, so here for instance, you look at one cell that is a stem cell, and then you look at all neighbor pairs, irregardless of what type they are, you see that after 2 hours, the fraction of neighbors that were still neighbors is still high, but it has decreased a little bit. And if you wait for longer, for 10 hours, you see that it has gone down further, but it's still relatively high. And it happens irregardless of the type of cell that you are. So it doesn't really seem to depend a lot on the cell type. So now we look not at any random neighbor pair, but we look at neighbors that are also sisters. Here what you see is that if you look at the different cell types, you already see that the fraction of cells that still remain neighbors after all that time is lower. But what is really striking is that if you look after 10 hours now, it's even lower, but it's in particular much, much, much lower for the secretory cells. And so what we think we see here is some sort of active demixing, that if you have two secretory cells next to each other, they tend to separate much more rapidly than any other type of neighbors. So what we think what is happening here is that that the patterning is not so much happening by not sickling as thought, but that instead what is happening is that the abundance is controlled by differences in proliferation, even though the number of progenitors that is generated is about the same, and that the patterning is then for a large part also established by this sort of mixing that is happening here. All right, that brings me to the end of my presentation, just a conclusion. So what I hope that I showed you is that, what makes organoids really nice is that you can use them to unveil dynamics, stochastic dynamics that is normally deeply hidden within our bodies. We can now do automated tracking of all cells. That gives us the position, it gives us lineage and movement. And if you then combine that with antibody staining at the end of the experiment, you can also integrate that with information on cell type. And that allows us, in the way that I've sketched, to get new insights in what sort of transitions are happening, how differentiation is happening, and how these patterns are established. And that brings me to the end of my presentation, and I'm looking very much forward to your questions. Thank you.